Hello everyone. I'm going to quickly go over our IDA assignment, which is mutation in the MDR1 gene. All right, so first step is to always read. So step one is to read the reading, okay? So reading states, the MDR1 multi-drug resistance 1 gene is found in dogs. So we have, this is our gene and it's found in dogs. This gene creates a protein called P-glycoprotein, which helps break down medicine in dogs' bodies after they are given it, okay? When the MDR1 gene is not functioning properly, okay, it can cause certain medications to build up or go into the brain and cause fatal reactions. This protein was originally discovered in cancer cells. When the MDR1 gene gets mutated, it causes dogs, specifically collies and Australian shepherds, to be more sensitive to negative effects of certain medications such as anti-cancer drugs. Okay, so though it doesn't tell us all the information, we can use what we've highlighted to help us understand what the MDR1 gene does. Okay, so we have our normal gene versus our mutated gene. So you need to then, step two, complete the protein synthesis, which is DNA to replicated DNA to mRNA to amino acid, for the normal gene first. Okay, so you would go through, if you don't know how to do this and you're like, oh my God, how is Ms. P doing this right now? You are going to go back into Teams and you're going to find the videos where it goes from DNA to replicated DNA, or DNA to mRNA, or mRNA to amino acid, so that you are not confused and you can see the step-by-step process. So when I'm turning my DNA into my mRNA, I always remember that A pairs with U and T pairs with A. So CUG codes for leucine, UCU codes for serine, and AAA codes for lysine. So now that I've completed step two, which is the protein synthesis for the normal gene, I'm going to compare my normal DNA with my mutated DNA and find the letter that's different. So when I look at these three, they're the same. So my first codon is the normal one. Second codon, the same. Third codon, the same. Fourth codon, I can now see that the A turns into a G. So in my fourth codon, okay, so this is my third codon, my second codon, and my first codon, I can see in the fourth codon, there is a mutation. Okay. So now I'm going to complete the protein synthesis just for this last one. So this is going to be step 3. Because you don't need to do the first, second, and third codon because they're identical to the normal gene. You're only doing the one that has changed. So now T pairs with A, C pairs with G, T pairs with A. And now I'm looking for AGA on my codon chart. AGA now turns it into arginine. So now I have a change in my amino acids. Okay. Once you've completed that, step 4, you're going to go down to your questions. Okay. So this is step 3. Now you're going to go step 4. And you're going to complete the questions. Guess what? Please circle where the normal gene and mutated gene are different. We already did that. That's right here. We highlighted it. Okay. Question number 2. Explain the difference between the normal gene and the mutated gene by looking at the above DNA, mRNA, and amino acid sequences. So they want you to compare both the DNA, the mRNA, and the amino acid. So when we do this, we can set this up: DNA, mRNA, and amino acid. So when we look at the DNA, that's our first row. Here's my DNA. How are they different? Well, we've highlighted the difference. The A turns into a G in our mutated gene. The DNA is different because in the normal gene, it's AAA, and it turns into AGA. Okay? You would then do the same thing for the mRNA. So when we look at our mRNA, same thing, AAA, AGA. So we could do this, which means it's the same sentence. So we can say the DNA and the mRNA. The DNA and mRNA are different because in the normal gene, it's AAA and it turns into AGA, right? The A turns into a G. Now, when we look at our amino acid, okay, originally it's a lysine and then it turns into arginine. So we're going to say that. We're going to explain that. The normal gene codes for the amino acid lys, and the mutation causes it to turn into arg. That's it. You are just stating what you have already noticed about the table when you look at these three pieces: DNA, mRNA, and amino acid. So that's question number 2. Question number 3, how will this mutation, how will the mutation affect the function of the protein? Okay, so remember, proteins are made from amino acid sequence. So if there's a change in the amino acids, that equals a mutation. So this is the normal protein. So we can say these 4 codons create the protein P-glycoprotein. Okay, so we can say, I would start off by looking at the reading. Okay, so normal protein. In the reading, it states, let's look. MDR1 genes found in dogs. The gene creates a protein called P-glycoprotein, which helps break down medicine in dogs' bodies after they are given it. Boom. That's what it does. That's what the normal protein does. So we can say normal protein, in the reading it states, P-glycoprotein helps break down medicine in a dog's body after they are given it. Done. Okay, so now I know what the normal protein does. So mutated protein, I am then going to go back up to my reading, okay? And I'm going to look at where it talks about what happens when it gets mutated. So MDR1 gene is not functioning correctly. It can cause certain medications to build up or go into the brain and cause fatal reactions. Boom. That is what happens. We've already highlighted it. You are using that from the reading to help you answer that. Therefore, you would then tell me,